So our next speaker then is Professor Dr. Julio Martinez-Trujillo. And Julio is a professor in the Department of Physiology and Pharmacology at Western, and also is the holder of the provincially endowed chair in autism studies from the Government of Ontario. Julio graduated from medical school at the University of Havana before completing his PhD at the University of Tübingen. Prior to moving to Western in 2014, Julio was an associate professor in the Department of Physiology and also Canada Research Chair in Neuroscience at McGill University. Julio's research interests are in the pathophysiology of autism, neurophysiological bases of attention, and also the neural mechanisms of working memory. And today, Julio is going to change gears a bit for us and talk about Rett syndrome. So, Julio.
Thank you very much, Rob. I'm going to share a screen now. Here we are. And you tell me if it is working. Is it working?
Yep.
And I actually would like to, oh, no, sorry. Let me just share the screen. And I believe that I need a laser pointer. So I have to confess that for this talk, I'm a little bit nervous because this is the first time that I talk about this data. And I really would like to thank Rob for introducing me to the autism community here, and has been really an incredible research partner. And organizing this thing has been really a great experience for me. So I'm going to talk today about electrophysiological characterization of human stem cell derived networks of excitatory neurons in the Rett syndrome. All right. So the Rett syndrome is some kind of a misfit for psychiatrists and neurologists, and I think that it has been in and out of the DSM-4. But the Rett syndrome, what is definitely is a clinical entity, and it has also a very good mapping from molecular geneticists and some of the people that study animal models. It's a postnatal neurodevelopmental disorder that affects females almost exclusively, about 1 in 15,000 females. It is characterized by normal early growth and development, followed by a slowing of development, loss of purposeful use of hands, distinctive hand movements, stereotypies, a slow brain and head growth. It has also problems with walking, seizures, and intellectual disabilities, and in some cases has symptoms of ASD. So one of the things that made us concentrate in the Rett syndrome in this specific work is that the Rett syndrome, it is known, is caused by a heterozygous loss of function mutation in the gene methyl-CpG binding protein 2, MECP2, which is actually located in the X chromosome. Now, to introduce to what we're trying to do here, which is basically link different levels of research, I probably should talk a little bit about how a brain is made in my perspective of a system neurophysiologist. So the DNA carried our genetic code for the making of proteins. And this DNA must be transcribed into RNA, and for RNA needs to be translated into proteins. The DNA alone is dead. The DNA alone is not a living thing, actually. It must be going through all these processes, produce proteins. And specifically in different tissues, it does different things, and proteins are different in different tissues. But specifically in the nervous system, some of these proteins attach to the cell membrane in a cell or in a neuron. And actually, some of them are ion channels that allow interchange of ions at very sharp temporal scales with the environment, in this case, sodium and potassium, and also the realization of the origin of the action potential, which is what characterizes the excitable tissue. So this action potential, of course, could happen in many neurons. Neurons could be different in the nervous system. There is not only one type of neuron, but many of them. And they are joined by synapses. And thanks to this action potential actually acting in the different neurons, there is a neural code that happens at this specific level. And this neural code in a whole brain produces behavior. This is the behavior the way that we know it and the behavior that many of the clinicians and behavioral scientists study. So all of these steps are important. This is something that I have learned over the years that I have been collaborating with James Ellis at SickKids. Now, the MECP2 is located in the X chromosome. And actually what it does is join this metal CpG binding protein, or MECP2 protein. It encodes the sequence for the MECP2 protein. What does the MECP2 protein does? Well, in the transcription of DNA to RNA, one can identify two different segments in the DNA. One is the euchromatin, where the chromatin is nicely aligned, right? And the other one is the heterochromatin, that the heterochromatin has like packages of DNA that is a little bit folded due to the existence of these proteins with several interactions called histones. Actually, the MECP2 proteins joins to these regions of the DNA and acts as a transcriptional regulator. So it's literally a master transcriptional regulator that allows the regulation of the transcription by, for example, repression or activation or compaction or chromatin loop formation. In this case, this process allows different cells express different proteins, even if they have the same DNA. So we know that the MECP2 regulates transcription and therefore protein expression. And the other thing that we know is that MECP2 is abundantly expressed in the brain, and it has been identified as normal in the Rett syndrome. I'm not going to refer to that. I'm not a geneticist, but there are many mutations actually of this gene that have been identified in patients with Rett syndrome with different levels of clinical syndrome. Now, our hypothesis is that the MECP2 mutations produce changes in the early development of excitatory networks in the brain of patients with Rett syndrome, and that these changes will be detectable at early stages of network development. So this is kind of important because what we're trying to do is to detect changes in the molecular actually makeup of the cell that produce functional changes that we can detect with the tools of system neurophysiology. For example, this is a synapse here, and this synapse is a very complex structure. So this is the presynaptic terminal, the postsynaptic terminal, and the postsynaptic terminal, we have NMDA receptors, AMPA receptors, and we have a whole machinery actually of enzymes. And we also have different, for example, transcription factors, or like in this case, the MECP2 seems to have an action on the expression of brain-derived neurotrophic factor. This is just an example. The functions of the gene in general, they have not been completely mapped. And we know that this has an impact in synapse formation in terms of NMDA receptor function, NMDA receptor populations at the level of the synapse. So considering this, we hypothesize that we may be able to detect changes in network formation when the cells are wiring in the nervous system in patients with Rett syndrome. Now, to do that, we use a model. The model is the human-induced pluripotent stem cells, or HIPSCs. And one of the advantages of these cells is that they preserve the complex genetic architecture and human developmental system. So basically, these cells could be obtained from humans, normal, or patients with Rett syndrome. And these cells actually could be a protocol that could be used. It's called the NGN2 protocol that regulates the commitment of neuronal or progenitor cells to the neuronal fate during development. So this is basically a transcription factor that makes the cells to go, okay, you are going to be a neuron now. So, and that happens through different transcription factors in different tissues. So with the NGN2 protocol, we can produce mutant and control excitatory neurons. And this is going to be the key to this project. So a history of this was the group at SickKids by James Ellis published this paper a few years ago, in which they look at SHANK2 mutations also associated with autism spectrum disorders, and they show hyperconnectivity in these human neurons. So, which is actually very interesting because once you differentiate those neurons and the neurons start wiring, you can do many things. You can actually do intracellular recordings. You can do anatomical characterization. You can do transcriptomics. There are many things that they did in this study. Now, how does it work? I'm not an expert on that, but I just as a general idea, you can take a biopsy or you can also use lymphocytes or different tissues, and you can reprogram those tissues. And actually you can induce pluripotent stem cells. You reprogram the pluripotent stem cells, and then you can induce different cell formation using different protocols. In this case, actually, the protocol that they used, it produced mutant and control excitatory neurons. Now, to get into the data, what happened, this happens in Ellie's lab, sick kids. So basically, they took cells from an unaffected individual, all from cell lines, human cell lines. They isolate the cells and they reprogram them, and they obtain the stem cells. And then they can use a CRISPR-Cas technology to produce a knockout of the gene of the MECP2, and then can produce knockouts, iPSC cells. Then you can take the wild type and the knockout iPSC cells, you pass them through the same protocol, and now you can obtain neurons with the mutation that have the knockout gene, and you can obtain also neurons that they're actually normal. So the wild type neurons, what I'm going to refer to as Rett syndrome neurons and wild type neurons or networks. Then you can put those on a plate, and this is a recording system from Axiom Biosystems that is basically a plate that has contacts at the bottom, very tiny contacts, in this case 64 for some of the plates, but there is a variable number of contacts. And then you make the cells to grow in those plates, and then the contacts can literally record the activity of these cells as they grow. What you will see is that after 3 weeks, or maybe 1-2 weeks, the cells start firing action potentials, and then over the course of the 3rd or 4th week, you see emerging actually activity that resembles or is almost identical to the activity in normal neurons, the way that we recorded it. You could also have potential to use, for example, tissue from Rett syndrome patients, and you can go through the same protocol, and then you can actually obtain these networks of cells. Now, what I'm going to talk about today here is basically a proof of principle that if you take these wild type cells and the knockout iPSC cells and you produce networks, you can find parameters that are indicative of the wiring of those networks, and then you can actually separate the wild type from the Rett syndrome. So this is how it looks, how a recording looks like. And basically what we have is here is a plate. And what you see actually coming in and out is basically the level of activity in the plate. What you see here is these lines that you see that appearing and disappearing. So they're action potentials fired for the different neurons in different channels. We're showing here, 8 channels in this case. And there are 64 channels in these plates probably. And these are 2 different plates. And you can see that the same thing is happening in the 2 different plates. And the other thing that you can see that if you build rasters, which is anytime that you see an action potential, you draw a vertical line, and you put them here aligned into the number of channels, you can see this pattern that is also, it's one of the most amazing things that I have seen, honestly. And it's this repetitive behavior that they're charging and discharging and discharging and discharging. And this happens at about a 3-4 week that these cells start doing that. Now, this looks very similar to the data that we record in system neurophysiology in the brain. So we have action potential, sometimes synchronic, sometimes not that synchronic. But what it seems to be that this is a marker that these cells are trying to wire together in this small network. So now we take this data from the Maestro plates. After we sort the data, there are here 4 different channels. And what you see here is trains of action potential, what we call a burst, which is a bunch of action potential wrapping very quickly, then stop, quickly, very stop. And you can quantify, for example, the interburst interval. You can quantify actually the burst duration, how long the burst is, how many spikes are in the burst. You can quantify the inter-spike interval within a burst, actually. And you can calculate other measurements like regularity of burst and different things. I'm going to talk about 2 or 3 measurements, because the whole idea is not to, so I have no time to go in detail over this, but the main idea to convey you is that this looks very aligned in 3 different channels. Remember, these are neurons that they could be very far away from each other in a plate, right? Several micrometers away, hundreds of micrometers away from each other on a plate. And these neurons are firing at the same time in this synchronic manner. That is, again, this is an impressive thing I have seen. So what I'm showing data here is data from wild type and from Rett syndrome networks. And here, for example, we have the burst duration. The average burst duration is higher in the Rett syndrome networks than in the wild type networks. Now, importantly, you have different colors here. It's a very pretty plot, but in reality, what that means is that these are different sessions that have been recorded across weeks from the same plate. And importantly, this plate has always a control plate. It's a wild type that grows in the same media, has been undergoes practically the same manipulations. Now, when you compute actually the average, you see that the Rett syndrome has a longer burst average than the wild type. In this case, we'll see, well, the Rett syndrome looks like it has a longer burst than the wild type. But if we look at measurements like, for example, the average interburst interval, what we can see is that the Rett syndrome actually has a longer average interburst interval. So the bursts are a little bit longer, but they're also happening at less regular time. So a longer space in between the bursts. And if you look at the coefficient of variation for interburst interval, for example, that is how regular the burst is. So what happened is that the Rett syndrome has a higher coefficient of variation. So somehow they're more space in time, those bursts, and they also have a higher variability in terms of when do they happen. So this is the first sign that we see, well, those networks, they don't seem to be actually developing in the same way in the Rett syndrome and networks and in the wild type. Now, a second measurement that we are very interested in is how, can we find a measurement of how these cells are actually networking together? They're going up and down together. And the second measurement is computed correlation coefficient between neighboring electrodes, and to see how correlated are those bursts that they happen in the electrodes. And here we have in blue the wild type and in red the Rett syndrome. And what you can basically see is that at week 4, the cells of the bursts start being correlated between different cells. Then in the wild type, they increase here. Basically the correlation between them. You can see this in these blue lines in the blue plot. So basically what you have, you see how the correlation start increasing until it reaches some certain plateau here. But in the Rett syndrome, they start a little bit early in this specific example. And then at week 4, they're kind of wired. But when you go to week 6 or 7, they kind of start declining a little bit, and they never reach the plateau of the wild type. So for some reason, those bursts are less actually correlated in the Rett syndrome than in the wild type network. Now, here is where Lyle Mueller's lab, and we started collaborating with that. And I think that we have now a dream team put together, Lyle Ellis' lab and our lab. And we started thinking, well, can we actually develop models of this? And Lyle said, yeah, sure, we can do that. So I got this wonderful student, Gabriel, and then Gabriel started working with Kartik, which is a student that have done a lot of this in our lab. And then they developed an integrate and fire model of a neuron, and by manipulating these adaptation parameters in this integrate and fire model, you can get this network to oscillate, or this neuron to oscillate and to produce a very similar behavior as we see in the Rett syndrome. In fact, when we actually look at the data, this is data collected from the plate, and I just, let me explain, this is the channel count, what you have here. This is 64 channels in the plate, and this is a magnification of the burst, bursting. You remember, those are rasters. Each one of these tiny bit of line is an action potential, and these are different actually electrodes in the plate, likely coming from different neurons. And this is actually the data, and this is the model. Of course, the model looks more prettier because actually they haven't introduced any noise on any other parameters that they can play with in the model. But the whole idea is that the model can replicate actually the data that was collected in the biological system. Now we went one step further, and we wanted actually to do a more strong quantification of this. And here we have, again, the recordings. Here we have the simulations in the model. And what you see here is basically a spectral analysis of this burst, of this activity. And what you see here, that there is these yellow peaks. So these are the different channels. The spectral analysis is done in each channel individually. And what you see, these yellow peaks here, is actually the dominant frequency, the nominant frequency of the burst. That's this large plot in here. Of course, this one are the harmonics of the frequency. And what you see here in red is the model, how the model actually approximate the main frequency and the harmonics of the frequency. What basically we have is a system that can be modeled. Now, we can also do the same thing with the Rett syndrome data. And when you do that with the Rett syndrome data, you have basically a similar thing. Here, the data, the spectral analysis for the data, and here, the modeling of the data. Now, the question is, and it's practically the second quantitative comparison that I'm going to show here, that we still need to do more on that, but the question is, are they different in the Rett syndrome and in the wild type? Are these dominant frequencies that the network is actually pulsating with different or the same? And this is summarized in this slide. We have here the peak frequency. And what you see in blue is the overall. If you pull all the data over weeks 2 to 7 of the development, what you see in blue is that the dominant frequency is around 0.3, between 0.3 and 0.4. But what you see in red here, which is the Rett syndrome data, is that the dominant frequency is below 0.2 hertz. So, and if you track them from week 1 to week 3, week 4, week 5, week 6, week 7, you see that in week 2, there is not much because the networks are not bursting very much. The neurons are not that active. But as you go in the different weeks, you start seeing that the burst, these histograms start being shaping as Gaussian distribution of somehow with a mode and a peak. And the differences start appearing between the wild type and the Rett syndrome. So as a summary, I don't want to give any conclusions. We can summarize that the development of functional connectivity in human iPSC-derived excitatory neural networks carrying MECP2 knockout, in this case, lack of expression of MECP2, is slower relative to wild-type control networks. This is concordant with developmental delays observed in patients with the Rett syndrome. The human iPSC-derived networks offer a window of opportunity to study early neural networks development in patients with Rett syndrome. They may also provide a model to develop interventions that can rescue molecular and functional alterations observed in Rett syndrome and other developmental disorders at very early stages. So I would like to say that the human iPSC-derived networks are an emerging model in neuroscience that can bring us closer to study human physiology and pathology. It has the potential to link molecular, cellular, and system neuroscience to study early stages of brain development. My last slide, this has been really wonderful experience for me, which is actually the collaboration with James Ellis. Sometimes we were drinking a coffee and we said, well, why don't we work together? And he says, I don't know how that is going to happen. I said, well, we'll make it work. And now it is working. Basically, you have James, Rebecca, and Fraser has been very important in this work. They have done a lot of work. And in my group, we have Kartik, that has been the heart of this in our group, Lyndon and Milat. And of course, you have Lyle's lab, where him and Gabriel have done an extraordinary job to model this data. Thank you very much, and I'll take questions now.
Thanks, Julio. That was wonderful. A lot of data in a short period of time, which was tremendous summary of your work. Are there questions for Julio? Something there, but somebody left. So Miguel Pena had a raised hand, but now it's lowered. Miguel, I don't see your hand. It was up, and now it was lowered again. Did you have a question you wanted to ask? while we're waiting for that, Julio, a question. So that, when you say that what you see is a sort of delayed development of connectivity in people with Rett syndrome, or at least in the sort of mouse model that you've developed.
That's correct.
Is there any relationship in the timing of that sort of development of connectivity to the sort of the slowing down of brain size increase?
Yeah, that is actually that we're not sure about that. It's an excellent point, because what we see is that after week 7, the cultures start declining just look at the connectivity slide here. If you look at the week 7, these things start going down. And what we see is that the cultures some of the cells start getting detached. It's complicated. So because you don't know, is what is happening is that the cultures are just dying, or that definitely there is some kind of regression going on in the networks, and the networks, they don't wire anymore. If you can see here, the Rett syndrome, this one is less connected than the ones at week 5 and week 6. So now how to map the time scales of this specific model into the time scales of development in an organism is very hard to do. I think that is in fact, I mean, if I have to confess to you that this is done with mouse astrocytes. Once you put human astrocytes in the culture, and once you actually start differentiating then into excitatory and inhibitory networks, we may see something different emerging from that. Something that may speak to that is the work done in organoids right now. So there are some people that are doing work on organoids, that those are maybe a little bit of a better model in terms of the 3-dimensional structure of the cortex and things like that. But it's hard to say, Rob. I would love to say yes, but I don't think that would be an honest answer.
Yeah, because that'd be really cool if there was some kind of relationship there. All right, Miguel Pena Ortiz has a question. So Miguel, if you could just unmute yourself.
Yeah. Hello?
Hello?
Awesome.
Yes.
Oh, great talk, Julio. Amazing data. I was wondering if, I think you mentioned the two that they've used this technique to look at the transcriptome and at the morphology of the neurons. I was wondering if you were planning on doing those kind of studies with your, with these same like plates that you have.
Yeah, the transcriptomics is not done. The morphology is done by James Ellis lab. So the neurons are smaller and they have fewer dendritic branches. So somehow the neurons have changes also in input resistance, membrane potential, and they also have changes in expression of sodium and potassium channels, the Rett syndrome neurons. Now I have to tell you that these neurons are full, actually, knockout of the gene. A lot of the mutations that you may find in patients with Rett syndrome are missense mutations that actually may produce more mild forms of the phenotype, of this phenotype. They did a study that actually is, we're writing up the study now with one missense mutation. And in this specific study, what we see is that the neurons still preserve this phenotype, that they're smaller, they have fewer dendritic branches, but the network seems to be actually less affected in terms of dominant frequencies than the full knockout. So I think that there is a lot to do in that respect, but yes, the neurons, they come at normal. Yeah.
That's very interesting, thank you.
So there's, I think Dr. Mitchell also had a question. Correct me if I'm wrong, Claire. So I've asked you to unmute yourself, and that should do it at your end.
No, I don't actually have a question. I just don't seem to have a mute unmute button for this whole thing. So I've been just waiting to have a question. But I thought it was a fascinating talk with so much potential for other neurodevelopmental disorders as well, to be able to study in that way. But it was very clear. Thank you.
But I think to that point also, though, Claire, I guess the question, Julio, would be, when Claire says it has the potential to be of value and import in a lot of neurodevelopmental disorders, I guess the question would be, what's specific to what? And I think that's a huge problem we have in a lot of research in neuropsychiatry.
Yes, that's correct. So the first thing that James, when we start doing this, I tell James, has to be completely messed up in terms of the phenotype. It could be really has to be a very strong phenotype for us to find something there. I mean, I came from the behavioral side where, okay, if you don't study behavior and your preparation not study behavior, then you not study the real thing. So I totally changed my mind. Everything is important. This is also very important. And what I can tell you is that probably the stronger the developmental deficits that you see, the more likely you're going to find those effects. So, and I also think that it depends on the protocols that they use. If they use protocols now that they can, for example, can differentiate and get cell type of specificity, for example, for amygdala cell types and things like that, we may be able to see things more in detail in those different things. But again, I think that the best candidates for this are these developmental disorders where you see a very strong clinical phenotype. Yeah.
Okay, thank you, Julio. That was wonderful. And we look forward to an update in the near future about your cure and solution for Rett syndrome.